All right, everybody, welcome back. We are going to really get into cell respiration. So we've already talked about overall what it is in the equation. We've compared it to photosynthesis. We've talked about energy. And at this point, you know that cell respiration is all about making ATP. That's the point. We're making ATP with this process. So we're going to learn really how cell respiration works now. And there are three main steps. There is glycolysis, that is the first step. And then after glycolysis, there is the Krebs cycle, followed by the electron transport chain called the ETC. We're going to go over the first two steps today, glycolysis and the Krebs cycle, and we're going to do the electron transport chain later. So cell respiration, depending on what organism it's in, it can be aerobic or anaerobic. The word aerobic, like aerobic exercise, it means that oxygen is in use. When we do cell respiration, we are using oxygen, so it's aerobic. Plants also have aerobic cell respiration, and they produce a lot more energy. But some things undergo anaerobic cell respiration, which does not use any oxygen and is not as good at producing energy, right? So not as much ATP is made. So we are really good at producing ATP because we have oxygen. Other things are not so good at producing ATP because they don't have oxygen. The very first step that we go over today is glycolysis. That's our first step of cell respiration. And glycolysis is the anaerobic part. So even though we breathe in oxygen, first step of cell respiration cannot use it. So it is an anaerobic step. So this is our first step, glycolysis, cannot use oxygen. Glycolysis is going to produce something that goes to the Krebs cycle. And then the Krebs cycle and glycolysis are going to produce things that go into the electron transport chain. That is the third step. All three of these steps are going to produce ATP. So step one, glycolysis. Glycolysis, know where it happens. It happens in the cytoplasm. So it is not in any particular organelle. It is out in that fluid of the cell. What happens in glycolysis? Well, overall, what's going on is we're taking glucose and we are breaking it apart into pyruvic acid. Pyruvic acid is what is left when glucose breaks apart. Glycolysis is going to use two ATPs. So ATPs are energy. So this actually takes a little bit of energy to get started. But notice what happens. It's going to create 4 ATP. So it's going to create more than it uses. So there's a net gain there of ATP, a net gain of 2. And it's also going to create 2 NADH. Hopefully we remember what that is from yesterday. What does NADH do? NADH is the electron carrier. It carries electrons around. So our net gain from glycolysis is 2 ATP and 2 NADPH. 2 ATP is a small amount of energy, and 2 NADH is a small amount of electron carriers. Remember that glycolysis, does glycolysis use oxygen? No, it doesn't. So it's not producing much ATP. 2 is not a lot. It's a little, we can use it, but it's not very much. It's not really going to keep us going in the long run. So this is how glycolysis works. This is a picture of the process. So let me explain to you what's going on here. So first of all, we begin with glucose, and glucose has 6 carbons. All right. Now, glucose is going to undergo a series of chemical reactions. And one thing that's going to happen is ATP is going to meet up with glucose. And what's going to happen is it is going to, kind of like workers coming to a construction site, it's going to meet up with a construction site, they're going to do a little bit of work and change it, and then they are going to leave. Well, ATP is going to meet up with glucose, it's going to do some work on glucose, and then the ATP is going to lose its energy, its phosphate, and it's going to go, it's ADP. So ATP is going to do work on glucose, and then it's going to leave. And what kind of work does it do? Well, it breaks glucose in half to 2 3-carbon compounds. So we have broken glucose in half. But we're not done. Those 2 3-carbon compounds are not finished yet, and other molecules are going to come along and they are going to do some work. NAD+ plus is going to come along. What does NAD+ do? Think about what we learned yesterday. NAD plus is going to pick up something. So hopefully you remember that NAD plus is going to pick up electrons. And that's what's happening. NAD plus is going to pick up electrons and become NADH. That NADH is going to carry the electrons somewhere else. So taking electrons away from those carbon compounds is going to change them. So they're getting changed once again. And then we have some ADP. The ADP is going to come along, do some work, and it's going to end up becoming ATP. So we're going to energize that ADP to make ATP. And it's going to happen again. And we're finally done doing all this work to the glucose, and we end up with 2 3-carbon compounds called pyruvate, also known as pyruvic acid. In the process of glycolysis, glucose breaks, we make ATP, and we make NADH in the meantime. So here is a shorter, so this is really what I want you to know for the test. This picture is showing pretty clearly everything you need to know. We've got glucose. Glucose is going to break in half to 2 pyruvic acids, or 2 pyruvates. And in the process, in order to facilitate that breaking, we are going to make 2 ATP, which is a little bit of energy that we can use. It's not much, but it's a little bit. And then we also make 2 NADHs, and those 2 NADHs are carrying electrons, can be taken somewhere else, and we'll find out where they go later. So this is really the important stuff to remember. Glucose breaks into 2 pyruvic acids, ATP. This is useful for energy. NADH, this is going to be useful later. All right. So let's take a look at a few questions here. What are the products and reactants of glycolysis? Include numbers. So talk about glucose, pyruvate, NADH, and ATP. And then also, where does glycolysis happen? And you should answer these questions now. All right, so the process of glycolysis, we start with glucose. And when we break a glucose, we end up with 2 ATP. And we end up with 2 NADH. And we end up with 2 pyruvic acids. And the answer to our second question, where does glycolysis happen? It happens in the cytoplasm. So if oxygen is not a part of glycolysis and glycolysis makes ATP, then why do we need oxygen at all? We just made some ATP. Well, the answer to that question is because we haven't made enough. 2 ATP is not enough. We are very complicated things. This might be enough for something very simple like yeast or bacteria, but we have a lot going on and we need more ATP than 2. So we've got more steps, and these extra steps are going to make more ATP. Some simple things like bacteria are not going to continue on. They're sort of done after glycolysis. So keep in mind, these next few steps can only happen if there's oxygen, which there is. If we are living, there is oxygen in us, and the same thing in plants. So if oxygen is present, the pyruvic acid is going to move into the mitochondrial matrix. That is the middle part of the mitochondria. So it's what I'm trying to highlight in red here. And the pyruvic acid is going to change and become a molecule called acetyl-CoA. So this is part of the respiration, part of cell respiration. Here's pyruvate. And we didn't draw pyruvate like this, but pyruvate has 1, 2, 3 carbons. So we have a 3-carbon compound here. And really the long and short of it that you need to know is that pyruvate is going to become a 2-carbon compound, right? You see how there's 2 carbons there? Acetyl-CoA has 2 carbons, and pyruvate has 3 carbons. So when we get to the matrix, the pyruvate loses a carbon. That carbon is lost to CO2. And you think about what you exhale. You exhale carbon dioxide. This is where some of that carbon dioxide comes from. And also, what do we get? We get a little NADH. NADH is going to steal some electrons from the pyruvate, and that's going to change the pyruvate a little bit. And then coenzyme A is added. We don't really care about that too much, but the net result is we end up with a 2-carbon compound called acetyl-CoA. And we got a little bit more NADH, and we got some CO2, which is basically a waste product that we're just going to breathe out. All right. And so then we are on to part 2 of cell respiration, and that is called the Krebs cycle. The Krebs cycle is also known as the citric acid cycle. It happens in the matrix, that is the inner space of the mitochondria. The Krebs cycle is going to use that acetyl-CoA that we just produced, and it's going to make a bunch of things. It's going to make NADH. You already know what this is. It's going to make FADH2. You also know what that is because FADH2 is just another version of NADH. It's slightly different, but does pretty much the same thing. Those two things are almost the same thing, just slightly different molecules. We're going to make some ATP, a little bit, and we're going to make CO2 as a waste product. So all of the products of the Krebs cycle are listed below. I don't really care about the numbers, so 8, 2, whatever, don't worry about that. I just want you to remember we get some electron carriers. Those are really important. We get a little bit of cell energy, not much though. It's 2 again, and 2 is a little bit, but it's about like having a $2 in your bank account. It's not really going to do much good. You need more than that. And we also have carbon dioxide, which is a waste product, and that's just going to get exhaled. So here is how the Krebs cycle is going to work. So here is our acetyl-CoA. This is what we just made. Let me get my pen back. All right, so we just made a 2-carbon compound called acetyl-CoA. Now already in the cell, there is a 4-carbon compound that's already present there. And what's going to happen is this 4-carbon compound is going to join with the 2-carbon compound. So 4 plus 2 is going to give us 6. So we end up with a 6-carbon compound called the citric acid, hence the name of the cycle. Now, this 6-carbon compound is really important, and it is going to undergo a series of changes. So that's what the bottom half of the cycle is for. All of these changes, the 6-carbon compound is going to go over. It's going to lose a carbon to make CO2. Some NAD plus is going to meet up with it. And think about what NAD plus is going to do. It's going to steal some electrons and make NADH. All right, so some ATP is going to get made. And think, the 6-carbon compound keeps getting changed all the time. More CO2, so we're losing another carbon. So now we're back to 4 carbons. We're making more NADH. We're making some FADH2, which we know is the same thing as NADH. And we end up at the end of the cycle with that very same 4-carbon compound that we started with. Another acetyl-CoA can join, and the entire cycle can start again. Let's look at another view of the Krebs cycle. So we have acetyl-CoA going in, and as the cycle goes around, as these tiny little chemical reactions continue, we make NADH, we make more NADH, we make FADH2, we make CO2, and we make ATP. So those are the main components of the citric acid cycle or the Krebs cycle. So what is the function of the Krebs cycle? The most important part is this that I'm writing a box around. It is making a bunch of NADH. I don't care about the number, it's a lot. We're making FADH2, and that's really important. We're also making a little bit of ATP. It's not much, so it doesn't really matter that much, but that is still useful to us. And it's making CO2, which is just a waste product. The really important part here is the NADH and FADH2. These things are carrying energy, and we're going to use them in our third step. They are going to go on to the electron transport chain, and they're actually going to power the electron transport chain. They have a really important job, which is why we've spent so much time focusing on NADH and FADH2. And that's where we're going to stop for today. Let me know if you have any questions.